Illinois Urban-Champaign. He received his PhD from University of Wisconsin, and he also was a postdoc at University of California. His research is actually focused on system and synthetic biology, and his research program are actually studying how cells process information, especially interested in understanding how decision-making process are encoded within individual cells through feedback control. He and his group are actually engineering bacteria and yeast to produce biofuels, and their goals is to optimize the uptake and metabolism of C5C6 sugar in bacteria by developing a new genetic tools. Dr. Chris, the floor is now yours, and we are really excited to hear about your talk and about your presentation. So the floor is yours now.
All right, thank you. So can everyone see my slides? Is that good?
Yes.
All right. So I'm going to in this talk, I'm going to talk about the regulation of virulence in kind of the model pathogen Salmonella typhimurium, or as it's now known as Salmonella enterica. And we've been basically looking at this as a model for really understanding complex gene regulation. And I'm going to summarize a lot of work. And it's probably about 15 years of work from a number of different researchers looking at Salmonella and trying to really disentangle the complex regulation. And at the very end, I'll talk a little bit about antibiotics and potential targets. This is kind of not really an active area in my group, but it's something we're starting to think about, and some other people have actually started to think about that. So before I get into kind of the science, I first figured, since it's an international audience, I'd show you where the University of Illinois is. If you can see my cursor, we're kind of smack in the middle of the country, kind of what's known as the Midwest of the United States. So most people know kind of the East Coast, like New York City over here, or Los Angeles or San Francisco over here, or maybe Texas down here. But we're in kind of the central Midwest part of the country, kind of, at least population-wise, kind of dead center in the country. Here's just a picture of our campus. This is kind of the fall season where the trees are starting to change colors. This is the kind of the central quad of the campus. Right now it's snowing outside. It's probably about 5 degrees well, no, it's probably about -5 degrees centigrade. And it's snowing, so it's not this beautiful, and it's kind of bleak outside. But you know, we're a big US university. We're about 50,000 students. We do everything that US universities do, including having large football games, though our football team's not very competitive. And we're kind of in the middle of what in the US in the Corn Belt. So we sit probably in 100 miles of corn in either direction. So Champaign County, where the university is actually the second most productive corn producing county in the United States. So we're a big agricultural university. Okay, with that, I'll talk about Salmonella. So Salmonella is a common foodborne pathogen. At least in the US, we have periodic outbreaks, probably 5 to 6 times a year. You know, it infects a couple 100,000 people getting food poisoning. It doesn't really kill too many people. It's more kind of infants and immunocompromised. So the number of deaths are kind of probably in the single or double-digit numbers. But worldwide, Salmonella, particularly when it goes to systemic phases of infection, like Salmonella, like typhoid fever, can kill, you know, well in excess of 100,000 people worldwide. So what I'm going to talk about is a lot about regulation, or how Salmonella kind of goes through the infection process. So the way it works is typically Salmonella is, as I mentioned, a foodborne pathogen. You eat contaminated food. It passes through the gut, where Salmonella has a pH stress response, so it can survive the stomach. And then it kind of does its invasion, or the infection process occurs in the distal part of the small intestine known as the ileum. And there's a couple of series of processes. So Salmonella is a motile bacteria, so it'll swim through the gut. And then it expresses lots of different pili that allows it to adhere to intestinal epithelial cells. And then it actually invades the intestinal epithelial cells. So most cases of Salmonella, if you're thinking about just enteric fever or just kind of self-limiting food poisoning, is just basically inflammation of the gut during this invasion process. So it upregulates various cytokines in the immune system, you know, activates kind of the immune response. You typically get neutrophils and macrophages attacking the site of infection, causing the leakage of interstitial fluid into the small intestine, which is the cause of diarrhea. And then the process repeats where the salmonella comes back, people eat it, and they get food poisoning again. In addition, salmonella can go into a persistence phase where it basically becomes quiescent and hangs out in the small intestine. So it's not pathogenic. It's not recognized by the immune system. And this is actually in the US is very common in cattle, where it goes in this persistence phase, and they tend to be a reservoir for salmonella, at least in our food supply. And then with certain strains of salmonella, in particular the typhoid strains or the typhi strains, salmonella can go to self-limiting to a more systemic level of infection by invading macrophages and circulating to the lymphatic system. And that's where you really kind of get the typhoid fever. So I'm going to focus on the initial phase of infection. And the way salmonella is able to infect epithelial cells is it uses a molecular hypodermic needle, which is called the SPY1. And I'm going to use this word a lot, SPY1, which stands for Salmonella Pathogenicity Island 1, type 3 secretion system. And basically what it enables to do, here's just a beautiful picture from Jorge Galan's lab at Yale University, where you see salmonella is expressing these type 3 secretion systems that allows it to inject proteins into the eukaryotic host. Those proteins commandeer the actin cytoskeleton of the host and promote engulfment of the bacteria within the cell. And that's really the infection stage. So once it's in the vacuole, salmonella upregulates kind of the immune system, and that's kind of the stimulation of kind of inflammation of the small intestine. So I'm going to start kind of before I get into the work of just the acknowledgements of who's kind of worked on this project. And so it's really, most of what I'm going to talk about is the work of 3 scientists kind of sequentially. So the first person who started this was a graduate student, Supreet Sani. He's now a professor at IIT Bombay. And then another graduate student, Santosh Koralia, took over. And then lastly, and this is kind of the connection with Egypt, is Helwan was visiting scientist in my lab, and she kind of has done kind of the most recent work in salmonella, and has actually kind of gotten me thinking a lot about kind of, using some of the knowledge we've gained, the systems biology knowledge, to really think about, developing antibiotics and targeting different processes. So let me just move on to the next slide. So just kind of the key question that I'm going to hopefully try to at least partially answer is basically how is virulence regulated in salmonella, and really focusing on that SPY1 type 3 secretion system. And just kind of an outline of kind of the subject I'm going to talk about. Since I'm going to talk a lot about gene regulation, and I don't really know the background if people have done a lot of molecular microbiology, but I'll give you a background on gene regulation. Then I'll talk about the regulation of virulence in salmonella. And then kind of where I think the more interesting questions are the connection of virulence with other cellular processes. I think this actually makes probably the biggest case for looking at, targets for antibiotics. There's also a key issue of phenotypic heterogeneity, which I'll discuss. And I'll finish off with kind of potential targets for antibiotics. So just kind of biology 101. And this is gene regulation in bacteria. So we can think about this black line as a strand of DNA. A segment of that is the coding region of DNA. This is the gene. And then upstream of that gene, typically 100 or 200 base pairs from the start site of the gene, we have the promoter. And this is kind of the recognition site for RNA polymerase. This is where it binds, depending on the sigma factor associated with RNA polymerase. And then you have transcription for mRNA, and then you have translation with proteins. So this would be kind of a case of constitutive expression. We can also have regulation of transcription. And what I mean by regulation of transcription is not all genes are expressed at the same time. So not all genes are being transcribed to mRNA. Not all mRNA is being made to protein. And typically what we have is additional proteins known as transcription factors that either, these are proteins that bind DNA that either promote or inhibit the ability of RNA polymerase to bind to promoter. So they can either be positive factors for transcription or negative factors for transcription. Where things get interesting is where we get feedback regulation, is where kind of the product of the gene, right, the protein being made is itself either a transcription factor or modulating the activity of the transcription factor. And what this does is actually creates a feedback loop where kind of the output of the system, being protein synthesis, is affecting the input, which is the ability of RNA polymerase to bind the promoter. Autogeneous regulation, which I'm going to talk a lot about, is when a protein product is a transcription factor. So the protein's being made and it directly regulates its own transcription, either positively or negatively. And then typically we also have an extra factor is that we have some kind of external signal that modulates the activity of this transcription factor. Typically the signal is the binding of a small molecule. Typically a metabolite will bind this protein and either turn on its activity or turn off its activity. And so a common one is negative feedback, where a transcription factor blocks its own transcription. So it prevents it, once it gets made, it kind of stops its synthesis. And then typically you have a signal that actually downregulates the ability of this to activate. So the signal actually induces the signal. And I'm going to, in subsequent diagrams, I'm just going to draw this simply of the transcription factor kind of inhibiting its own transcription. And here's just what typically enables this. What this does, this kind of negative feedback, is it gives you very tunable expression. So it kind of gives you a nice kind of smooth curve. Here's just a system where we're looking at gene expression. And in this case, we were looking at a protein that binds tetracycline. And as you can kind of increase the tetracycline concentration, you kind of increase the expression of that protein. And this is kind of a negative feedback connotation. We can also have positive feedback, where the transcription factor is expressed and then it activates its own expression. So kind of you have this positive feedback loop. So once you get a little bit of protein, it can kind of take on itself. And typically what happens in a system, here again, is just playing with that tetracycline example. If you add positive feedback, you typically amplify the response. So, often we think about amplifiers as involving positive feedback. This is kind of a sound amplifier, but it happens the same context in the cell. Where things get really interesting is that you rarely see a single transcription factor by itself. It's typically interacting with all different kinds of transcription factors. So you get these kind of complex networks of interacting transcription factors, where maybe you have one transcription factor that's responding to the signal. It can activate its own expression, but it can also activate other kinds of transcription factors that can also activate their own expression. And then typically these converge on a downstream target gene. And it doesn't necessarily have to be negative feedback. It can also be some negative feedback. It can be positive feedback. So here would be positive feedback. Here would be negative feedback. Maybe this one activates this transcription factor and this one inhibits this transcription factor. So you can get this complex circuitry within the cell. So that's typically the model or the analogy that we use, that we think about these transcription factors as almost like circuits, components in an electrical circuit. And this is where we get logic and kind of the information processing that goes in the cell. So, even though these are single cell prokaryotic microorganisms, they have simple brains that allow them to kind of understand their environment and make decisions based on the signals that they perceive in their environment. So one of the things I've always been interested in is what are these interlocking feedback loops doing in the cell? What is the logic? What's happening with all these different transcription factors? And how do they enable cells to kind of process information and make decisions? So one other thing in the tutorial, I'm going to talk a lot about measuring gene expression, and there's lots of different ways to measure gene expression. So some of you might be familiar with microarrays and RNA-seq. And this is just a way to kind of get a snapshot of all the mRNA being made in the cell. At the very end, I'll show you one kind of RNA-seq experiment we've done in Salmonella. But primarily, we don't actually use microarrays or RNA sequencing. We actually use transcriptional reporters. And the reason why is that these are much more quantitative and allow us to look at the behavior of cells kind of at single cell resolution, what I'll get to in a second. So the trick we do, and we take advantage of genetic engineering here, is we have kind of a native gene. So we might be interested in some gene in Salmonella, say a virulence gene, and then we have its promoter. And so what we do is we actually clone the promoter at a different site in the chromosome. Come over here, and then come up over here, and then downstream of that promoter, we put a reporter gene. So we basically copied this promoter. So whatever's affecting this promoter, which is going to drive the expression of this gene, will also drive the expression of the green fluorescent protein. And once we have green fluorescent protein, and we've used other ones such as beta-galactosidase, which you can add a chemical and it changes colors, or luciferase, where we produce light, is that it gives us a signal that we don't actually have to open up the cell to look at. and we can kind of get real-time, quantitative, dynamic data from the measurement of this reporter system. And so basically all the data I'm going to show is using different kinds of reporters. It doesn't really matter which one, the details would be on our paper, and I'm happy to discuss it afterwards why you would use one reporter versus the other, because they all have strengths and weaknesses. All right, so I mentioned that kind of the key process involved in kind of virulence in Salmonella is the expression of this type 3 secretion apparatus called SPY1, or the SPY1 needle complex. Again, it's a molecular hypodermic needle that takes proteins from the Salmonella and injects it into the eukaryotic host. Well, it turns out this needle complex is regulated by a single transcription factor called HIL-A, and HIL-A stands for hyperinvasive locus A. So when they were doing genetics back in the early 90s and they were mapping these genes, this was actually the first gene they found, or the most significant gene they found that affected virulence in Salmonella. Turns out that HIL-A, which is a transcription factor, in turn is activated by another transcription factor called HIL-D, and that responds to different kinds of environmental signals. Typically, those environmental signals are short-chain fatty acids, osmolarity, and low oxygen conditions. And that's kind of the way we grow our cells to kind of induce virulence in vitro. But what's interesting is HIL-D activates two other transcription factors, HIL-C and RTSA. Environmental signals also induce the expression of HIL-C. All these transcription factors auto-activate each other. So HIL-D can induce its own expression, HIL-C can induce its own expression, RTSA can induce its own expression, and then they all cross-regulate one another. And then you have a repressor, which is HIL-E, and I'll return to this a little bit later at the very end of the talk, that actually binds HIL-D and prevents it from activating this cascade. And HIL-E responds to some internal signals in the cell. We don't actually know what those are, but we have a sense that at least one of it is kind of the fimbral system will activate HIL-E, but beyond that, we don't actually know what the signals are for it. So if you look at the expression of these different genes, so if you look at the expression of HIL-A, HIL-C, RTSA, HIL-D, and here we're just kind of growing it artificially in conditions. Salmonella is just grown in a test tube. And basically, we just fill up the test tube. We try to minimize the amount of oxygen. We grow them just in LB media, which is kind of common growth media, with a lot of extra salt. And actually, we can see induction of the different genes. So here would be HIL-D, which is the master regulator, HIL-C, HIL-A, the activator. And so they all just kind of turn on at roughly the same time. Maybe you can argue HIL-A is a little bit downstream. It's activated a little bit differently. But it just kind of, this circuit just kind of turns on and kind of activates the expression of the virulence genes. What's interesting is if you delete, so we can do genetic knockouts, we knock out HIL-C and RTSA, they actually have only a minor effect on HIL-D and HIL-A expression. So maybe you see a 20% reduction in the amount of gene being expressed. So they seem to be tuning expression rather than having a really strong effect on kind of expression of the needle complex. But interestingly, if you take out HIL-D, which is at the top of this cascade, you basically turn off expression of HIL-A. So HIL-D is absolutely necessary. HIL-C and RTSA are not. If I'm not going to show the data, but if we knock out HIL-D, basically this curve would kind of shoot up, kind of up here, kind of doubles the level of expression since HIL-E is a negative regulator. So again, arrows are positive actions. This kind of block area is a negative action. So when I first started this project, we knew a little bit about kind of this cross-regulation, and I was working with a microbiologist in Illinois, Jim Slough, and kind of the question is, what are all these loops doing, and what is the function of the circuit? You know, what's happening with this? Can we work out the logic? And so this is going to be a little bit more of an initial part on the systems part of the biology, and then I'll get a little bit more into kind of the physiology and some of the more complexity of the circuit. So first thing we did is we actually constructed a simple computational model of this circuit. And I won't go into the details of the computational model, but what the computational model does is we kind of took all our data, focusing on this induction period in the gray box. We kind of matched the characteristics on a computer. And then what the computer allows us to do is, at the time when we started it, we didn't really have a lot of ability to control the signals inducing the expression of different proteins. It was basically, we just turned the system on. We knew what turned it on, but we couldn't do any fine tuning. But once you get in a computer, you can start playing around and just changing numbers and kind of getting a sense of what, the sensitivity of the circuit is to the expression of different proteins. And what we found out is if we think about the signal, that basically this circuit encodes a threshold mechanism. So what I mean by the step input is the strength of this signal, and this is just a computer model, right? So it's not reality yet. But what we predicted was that when this signal is below a certain magnitude, and these are arbitrary units, we don't have absolute concentrations here, that there's really no expression. And then once we exceed a certain threshold, we actually just kind of turn on expression. It's almost like a light switch, or, once the signal gets, past 0.22, at least on the computer, we just kind of activate expression and everything turns on. And this is a little bit, this kind of switch-like behavior is a little bit weird for biology, because normally we think in terms of continuous concentrations, but all the complexity of this feedback loop kind of gives us this behavior. And I'll explain how it happens. And so one of the key things we found in the model was that actually HIL-E, this repressor, actually determines the magnitude of this threshold. And that was probably the most important thing from our analysis. And so the basic idea is, in the absence of positive feedback, if we're just thinking a linear chain of HIL-D to HIL-A, we have an input, and then we'd expect the output to be, in this case, kind of expression. Input would be HIL-D expression, output is HIL-A expression. We expect it to be proportional. When we start adding all this positive feedback in, we actually kind of increase the slope of this response. So we make it more sensitive, so to speak. And then when we get this repressor here, this HIL-E repressor, we basically put this threshold mechanism and kind of translate this kind of sharp response curve to the right. And so that means when the input's below this threshold, we get no expression. And then once we kind of pass that threshold, we activate it. Easiest way to think about this is HIL-E binds to HIL-D, and there's only a certain amount of HIL-E made to the cell. So for HIL-D to be free, we actually have to make more HIL-D than we have of HIL-E, because HIL-E will just bind all the HIL-D and inactivate it. And so that's really where the threshold response comes from. And so then the question was whether we could test this prediction. And so the way we did this is we knocked out HIL-C, and we replaced a HIL-D promoter with a HIL-C promoter. And the reason why we did this is we knew the HIL-C promoter was much weaker than the HIL-D promoter. So we're weakening the signal here. And when we looked at the data, here would be the wild type response of HIL-E. When we kind of replaced this promoter, right, so we have a weak signal, we got actually no expression. So basically we're below that threshold for activation. And then once we knocked out HIL-E, we kind of see that activation. And this kind of gave a rough kind of proof of principle test of this kind of threshold mechanism. But this still doesn't explain what RTSA is doing, what that HIL-C over here was doing. And this is kind of pure speculation. And again, we can only look at the computational model to get this. We don't have the fine control, at least with experimental data. But if we look at that top regulator, HIL-D, and we look at its expression, we see a huge change, right? As we increase the primary loop, which is HIL-D, you see it kind of rapidly increase on the expression. Here's the threshold mechanism. But we look at those ancillary loops, the ones on the side, so just go back, this one over here and that one over there, we see kind of much more fine-tuned expression. And kind of the basic analogy we think about this is that the ancillary loops kind of just fine-tune the response. And the analogy I think about is a microscope, where you have kind of typically two knobs. You have the coarse knob, which would be the primary loop, which kind of gets you in rough focus. And then you have the finer knob, which kind of gives that fine-tuned resolution, and you can really see the crispness of the response. So that was kind of the idea when we first kind of looked at this response. And this was kind of basically our first paper on the subject. Now, once we start thinking about SPY1, we start thinking about all these other steps in regulation. So you have motility, you have adhesion, and then you have persistence. And I'm not going to talk about adhesion, but I'm going to talk about motility and it's also the connection to persistence, kind of on this branch of the loop rather than kind of the systemic phase of infection. So it turns out there's a lot of additional feedback loops. So one of those is the feedback circuit regulating flagella, and I'll talk about this more in a second. And flagella are these like long appendages from cells. They're typically about 5 to 10 nanometers in length. microns in length. So typically bacteria is 1 to 2 microns, and they run really long. And you can think about them as long arms on the cell that allows it to swim in liquid media and also swarm over surfaces. It has an equally complex network. And then there's also another one which is involved in the type 1 fimbriae. This is the adhesion proteins, or like Velcro on the cell that allows it to persist in the small intestine. And it is also regulated by 3 transcription factors, which I'm not going to really talk about this network too much. But what we were thinking about was kind of just the steps in, kind of invasion, right? So we have first motility. The cells have to swim to the site of infection. Then they have to kind of bind to the intestinal epithelia. Then they have to promote kind of the expression of the invasion genes. And then they have a decision point. So one is whether they kind of get inside the cell and they go to the systemic phase, and that means they go to intercellular pathogenesis. And then they activate a subsequent program, which I'm not going to talk about, or they go to this other branch where they go to intestinal persistence. And the nice thing about this one is that we can do this totally in a test tube, right? We can mimic the conditions of the small intestine. And so when you go in a test tube, basically cells will go through this kind of decision tree over here to the right, where they go from motility, adhesion, invasion to persistence. Let me get to the next slide. So it's kind of interesting from my perspective, if you look at the dynamics of gene expression, so here we're using transcriptional reporters, and if you look at the expression of the flagellar genes, you see those turn on first, and then they kind of start turning down. And then you kind of see activation of kind of the invasion genes. They kind of turn on and then turn off. And then only as the cells kind of stop growing do you see kind of activation of the fimbral genes involved in type 1, involved in intestinal persistence. We've actually looked at all the gene regulatory networks for these. So this would be the motility genes. This is the invasion genes. This is the fimbral genes. I'm sorry, I don't match these colors. But it turns out that a lot of the regulators for invasion regulate motility genes, regulate invasion genes, and all different kinds of genes regulate the persistence genes. And you kind of work out this kind of regulation where flagella activates invasion. But once you start invading, you kind of turn off flagella. Likewise, when you're trying to move, you're not trying to adhere to cells, so they mutually repress one another. And then once cells go into persistence, they also kind of repress the virulence genes. And you kind of get this simple logic again from like movement activates invasion, but once you're invading, you want to repress movement. Persistence represses invasion, and then persistence and movement are mutually exclusive with one another. So they also repress one another. Now, when we were thinking about this, we were also thinking that, this network over here, let me go to the next slide, is, what about the dynamics of these other networks? How do they affect the regulation of virulence? And in particular, the network I've spent the most time on is the one involved in flagellar biosynthesis and motility in Salmonella enterica. And so just a quick background, bacteria move, right? They don't always move, but under certain growth conditions, they move. Here's just a droplet of cells swimming under a microscope, and you can see them swimming around. They swim about 10 body lengths per second, and they're pretty modal, and they can move around really well. At the base of it, what enables them to move is what's known as the flagellum. It's a rotary motor. So this is the flagellar filament that I showed you an image of. And basically the way it works is by spinning that filament using a motor that's about 40 nanometers in diameter. It spins about 300 rotations per minute. You can actually monitor rotation. So if you grab the cells by their filament, kind of like grabbing a dog by its tail, and at least in American vernacular, we have a term called when the tail wags the dog, right? And so what you can do is you can actually grab the filament, and if you hold it, actually the cells will spin around with one another. So we were interested in working out, like, what is this network doing? Why is it so complicated? Why do you need so many transcription factors just to turn on expression of the flagellum? And all of these are just involved in activation. There's other ones involved in the sequential building of the flagellum, which I'm not going to talk about. And so the key observation is, and so let me step back a second. So in order to explain what it's doing, I have to emphasize a certain key point in gene expression. So there's two ways gene expression can occur. So one is it can be continuous or it can be heterogeneous. So here, typically we have a signal, like as we activate a signal, sometimes it's just time is that signal, is that you can have all the cells behaving the same. So initially, none of the cells are expressing a gene. As the signal increases, the cells express the gene maybe at 50% maximal expression. And then once you saturate the signal, they're fully expressed. But you can also have a heterogeneous response rather than this kind of tunable where all the cells in the population are behaving the cell. Rather, the signal actually turns on the fraction of the cells expressing the gene. So at intermediate signal strengths, some cells are not expressing the protein where other cells are expressing the protein, and they're also expressing it at kind of the maximal level. And then as you increase the signal, basically you get all of the cells to express the signal. And this is what we typically call heterogeneous gene expression. Other terms people use are bistable gene expression and phenotypic heterogeneity. And it's an interesting thing because it generates non-genetic diversity in a cell. So it's not encoded in differences in kind of the genome sequence. This will happen in genetically identical cells. It's entirely mediated by kind of gene regulation and interactions between transcription factors. And the way we measure this, and I'm going to show you a lot of plots, is that we use flow cytometry. So here's just a microscope. Here's a cell expressing fluorescent protein. And you can see that individual cells will express different levels of intensity. So this is kind of a heterogeneous cell population. And basically what flow cytometry does is we take a liquid culture of cells expressing a fluorescent protein, which we can activate using a laser, and then measuring the fluorescence using a photomultiplier tube. We take a liquid culture, we dilute that liquid culture, typically in phosphate buffer saline, and then we pass it through a device known as a flow cytometer, and we dilute it enough that when the cell kind of passes through the laser, we only on average have one cell at a time. And so we can count events, and we get these histograms where the y-axis is the number of cells and the strength of expression is the x-axis. And in a homogeneous population, right, if I go back to this homogeneous population, you do FACS, typically you get like a Gaussian distribution. Not all cells behave the same. Some will express it a little bit higher, some will express it a little bit lower, but they kind of have a peak. And, when we measure bulk levels or mean expression, it's typically associated with the mode of the distribution. However, in a heterogeneous population, like over here, what we typically, and let me slow down, I went a little bit too fast. And then as we increase this level of this in a homogeneous population, as we increase the strength of the signal, we typically shift this Gaussian distribution or this thing that looks like a Gaussian to the right. So the mean is kind of increasing in the population, but we still express some variability. A lot of this is just due to the timing of where cells are in the cell cycle. Some of the higher level ones are cells that are getting really big, that are getting ready to divide, and these might over here be newly divided cells. However, if we consider the heterogeneous case, we get these kind of bimodal peaks where we have one peak and then typically another peak associated with expression. The one to the left, we associated with the off state. So that's where cells are not expressing the protein. So all cells will exhibit some level of fluorescence. And then what happens as we increase the signal strength is that the population in the on state, off state decreases, and the population in the on state increases. So we kind of get these like multimodal peaks or two kind of peaks in the expression level. And you're going to see a lot of these plots in a second. And this was just to kind of enable you guys to kind of get a sense of, what these curves look like. Because it can get kind of confusing, and I'm going to start stacking lots of these on top of one another. So one of the first things we did, and this was kind of an early experiment, is that we kind of grew up Salmonella, and we initially grew it in a middle media, and we were trying to count the number of flagellum that the cells had. And we were actually finding that a lot of cells didn't have flagellum at all. And I actually thought my student, and this was Santosh at the time, was actually shearing off the flagella when he was preparing the cells to look under them under an electron microscope. That's where you can actually see the structures. And he kept on doing the experiment. He's like, Chris, I'm not shearing off the flagella. And it's like, let's just look under a microscope. And lo and behold, the minimal media that we're using, we actually saw most of the cells were actually not moving. They're kind of drifting about, and only a handful are actually really moving. But then interestingly, when we switch to nutrient-rich media, we actually see most of the cells kind of moving around. So this told us that we could actually tune kind of the fraction of cells using nutrients, or how nutrient-rich the media is. And the way we do this is using yeast extract. And there's not a single chemical in the nutrients that induces motility. It's actually just a general nutrient state to the best of our knowledge. You can actually see the tuning. So here we're just varying the percentage of yeast extract in the media. The white refers to non-motile cells. The blue refers to motile cells. And as you increase the fraction of yeast extract, you increase the fraction of motile cells. So this is kind of what we call phenotypic heterogeneity, or a bistable response. And you can see it on the level of gene expression. And here's these kind of flow cytometry plots where here's the strength of expression, here's the number of cells, this is just normalized, and here is kind of what we're doing is the strength of yeast yeast extract. And so here's a negative control. Here's a cell expressing no fluorescent protein, and this is just the background level of fluorescence that any living organism has. And you can see it's like a Gaussian type distribution. When we grow the cells in no yeast extract, we see basically the same response. So it looks identical to cells not expressing our fluorescent protein. But then as we go to intermediate nutrient concentrations, we actually see two peaks. So we see one peak associated, say, like 0.2. This big red peak over here is correlated with the white non-motile fraction. And then this peak over here is correlated with the motile fraction. And then as we increase the yeast extract concentration, the fraction of cells that are non-motile decreases and the fraction of cells that are motile increases. But we always have a small population that are always non-motile, and we've actually never figured out what's causing this population over here. So there's always like a non-motile population. Here's just a time-lapse microscopy image where we embedded the cells in agar using fluorescent protein, and you can see some cells are growing, expressing the fluorescent protein, other cells are not, right? And here we get again, and then eventually the cells will photo bleach out. But what the point of the video is, and this is really not anything that informative, is just to show you that we're growing cells. Some are expressing the fluorescent protein, in which the case is associated with flagellar gene expression. Other cells are not, even though they're all being grown from a genetically identical population. So our question was, what element in the circuit kind of regulates this heterogeneous response? And this is a lot of work, and I'll just kind of summarize it. It turns out there's two regulators, Phi-Z and YDIV. When we knock out Phi-Z, when we delete it, basically we get a homogeneous response to yeast extract. And we knock out YDIV, basically all of our cells are kind of in this motile on population. And so this kind of puts everyone motile and makes it non-responsive to yeast extract. It turns out nutrients regulate YDIV. And basically the way you can think about it is we have these two regulators, YDIV, and they mutually repress each other's expression. So when YDIV levels are high, Phi-Z levels are low, and Phi-Z levels are associated with expression of flagella. And so this would be the off state. When Phi-Z expressions are high, they repress the expression of YDIV, which allows flagellar gene expression to occur. And it turns out that nutrients repress the expression of YDIV. And so we have two mutually repressing or antagonizing regulators. Only one can win, and the off state, YDIV wins, and the on state, Phi-Z wins. And so that's kind of the short story. And so one of the questions you might ask is, why heterogeneity? And the way I typically think about this is in bet hedging. So if you were going to gamble and play a roulette wheel, right, it makes no sense to put your bet necessarily on just one number. You might want to spread out your bets on lots of different numbers because you don't know what the spin will come up to. It's kind of like investing. If anyone invests in stock, you don't want to put all your net worth into a single stock. You want to kind of branch it out. Cells do the same. They don't really know what kind of environment they're going to be in. And so it turns out that some will express a certain behavior where others will not. So motility is a classic example of phenotypic heterogeneity. that only only a fraction of the cells will become motile and the other fraction don't. And the argument for it is that when cells express flagellin, which is involved in the filament, this is sensed by the innate immune system in the cell. So it's sensed by toll-like receptor 4, and it kind of activates a really strong immune response, which sometimes Salmonella wants. So some of the cells want to be invasive, but they'll get cleared by the immune system, but the other ones want to be non-invasive, and then they can be kind of more quiescent in the cell. So that's kind of typically the argument for heterogeneity. So the question we're asking, and this kind of comes to Helwan's work, is we know that the flagellar gene expression, already talked about these kind of mutually YDIV and FlyZ and the nutrient regulation. We know that this has a heterogeneous or bistable response, but we know that flagellar gene expression is tightly coupled with virulence gene expression. And we're wondering whether we'd also see bistability in virulence gene expression when we see bistability in SPY1 gene expression. And around that time, we also got a new signal. And so we actually learned how to modulate SPY1 gene expression. And it's basically what we're using is acetate. So I'm going to talk about two signals. So one is using the short chain fatty acid acetate, which is the cell's sense as a signal for being in the small intestine, because acetate concentrations give them the millimolar concentration, and then also tuning nutrient concentrations. And so here is just kind of example. Here's looking at SPY1 gene expression. And here we're just increasing the yeast extract concentration or the nutrient concentration. And basically, when we grow it in normal media that would induce flagellar gene expression, we see basically all the cells in the off position. But then we add different kinds of inducers of virulence gene expression like salt. What we see is we start seeing most of the cells are in the off population, but we start seeing some cells expressing the virulence genes. And then when we add acetate, we actually see now two populations, one kind of a virulence, one expressing the virulence gene and the other one not. And the amount of nutrients in the growth media affects what fraction the cells are in the off or on state. And this was kind of interesting because we're seeing that it was really kind of, I'm not going to go through the details, but it was really that bistability in the flagellar gene expression was inducing kind of this heterogeneous response in virulence gene expression. One of the interesting things, and just a little bit of biology, and this will kind of get into kind of the finishing part on antibiotic resistance, is that we knew that other groups had found out that short chain fatty acids can activate virulence gene expression. So they're kind of sensed by HILD. And we also showed that nutrients can induce kind of virulence gene expression. So here we're just growing in a basal media. This is tryptone broth. And as we increase the yeast extract, this is basically LB, virulence gene expression, we kind of see an increase just due to kind of the nutrients. And this is solely due to when we also add acetate, and then we're getting much more gene expression. Of course, you have to understand this is kind of a bulk level measurement where we're measuring, a whole milliliter of culture. At the single cell level, what we're actually tuning is the fraction of cells expressing the flagellar genes. It's just this is a little bit easy to activate. But this was kind of the last experiment that Helwan did, and she just published a paper on it last year, is that interestingly, when we knock out HIL-E, we actually see no activation by short chain fatty acids. So this would be the wild, this is again a cell lacking HIL-E. So before we see activation, it's actually higher than before. So over here would be the wild type. We don't see a lot of activation by yeast extract. And here we see a lot of activation. But then we made the interesting observation that when we added acetate, basically expression was completely repressed. And as we increased the amount of acetate, the expression decreased. And so Helwan, this is where we did a lot of RNA-seq experiments because we had no idea what the mechanism was. And so this is kind of a heat map of looking at all different genes in a snapshot. And key thing is that blue refers to lower levels of gene expression and red refers to high levels of gene expression. And here's just the wild type. And here's in that wild type where we knocked out that HIL-E and we had yeast extract and acetate, and basically everything shuts down. All these other conditions, we're actually seeing activation of the virulence genes, but this weird protein of HIL-E over here kind of downregulates the expression. And we're actually, when we looked for a synonymous case over in this column over here, we actually found that it was genes involved in envelope stress, interesting, which is the RCS system in Salmonella. So this is membrane integrity, is strongly upregulated in this mutant. And it's known that envelope stress represses virulence. So somehow the loss of HIL-E is affecting kind of the stress response to the membrane in Salmonella. We never able to figure it out, but when we knock out kind of this regulator RCS, basically it enables, in the absence of HIL-E, for acetate to still activate virulence. So that was kind of just an interesting side bit of biology. So what about antibiotics? And this is where I'm going to finish. So one of the experiments, this is probably the last experiment someone did, is we looked at the effect of N-acetylcysteine, or NAC. It's kind of an antimicrobial. It's also a common kind of over-the-counter nutrient supplementation. And we found that the addition of NAC, which inhibits growth of Salmonella, actually inhibits the expression of the virulence genes and the motility genes at sublethal concentrations. But probably what the coolest thing was, and we never actually figured out the mechanism for this, is that we decided to look at antibiotic resistance. So we knew if we grew cells in the presence of sublethal concentrations of NAC, here's just kind of the growth curve, and we went for about a month of repeatedly growing in the presence of NAC, the ability of NAC to inhibit growth was basically lost. So the cells were no longer inhibited, and we saw kind of robust wild-type growth in these kind of resistant strains. We actually sequenced these strains, and we were actually never able to figure out what the mechanism was. But what was cool was that even in these evolved strains, so ES revolves to just ES1, 2, 3 is just different clonal isolates that were evolved in the presence of NAC. Some got more resistant, some got less, is that they were all attenuated for virulence, even in the presence of, so here would be kind of, I think I got the wrong picture. Oh yeah, is that they were still kind of attenuated for virulence, even in the presence of no NAC. So it kind of shut down kind of expression of the virulence genes. Don't know what the target is, but it seems that, you know, maybe it's targeting the virulence. That also means that the motility genes are repressed, or maybe it's more likely repressing the motility genes because motility has a stronger effect on virulence and vice versa, that we're repressing the motility genes, and that in turn is causing the repression of the invasion genes. And just kind of a last plot. This is not my work, but this is work from Lon Chubis, who's a former graduate student in my lab. And when he was in my lab, he was looking at kind of the innate resistance mechanism in enteric bacterium, particularly the resistance to salicylic acid and paraquat, which again are two antimicrobial. Salicylic acid is common, it's very commonly used for antibiotic regulation. And one of the last experiments he did in my lab is he showed that salicylic acid strongly inhibited virulence in Salmonella. We had no idea what the mechanism was. Lon went off to a postdoctoral fellowship and then started his own lab in Missouri. And basically he showed that salicylic acid and also this other regulator paraquat actually repress the motility genes in Salmonella. So it has basically its effect on virulence is through basically repressing the motility genes. And this makes it look like that motility might be actually a good target to go after to also repress virulence, at least in kind of enteric pathogens like E. coli and Salmonella. So with that, I'll conclude and say that regulation, hopefully I've explained this, is a really complex process involving many genes and transcription factors. I've provided just a snapshot of that complexity. There's lots of other labs working on it, and there's lots of other processes that are coupled with virulence. Our interest is really the coupling of virulence and motility. And I think that, this gives us the ability to target, think about antibiotics, maybe not necessarily killing the cells, but attenuating virulence and motility in those cells, preventing them from being infectious, such as N-acetylcysteine. And, while a lot of the molecular details are known of this regulation, we still have many questions regarding what kind of the specific role of what these regulators are doing. So why all the complexity? What's the logic of this regulation? And then the last question we haven't looked at is how does heterogeneity affect resistance and virulence? If an antibiotic is targeting motility in a cell, but only 50% of the cells are motile, what about the non-motile cells? And there's a lot known about persisters, non-growing bacteria that kind of resist antibiotics. Potentially heterogeneity could give cells another mechanisms for resisting virulence. And with that, I'll conclude. Here's just kind of an old picture of my group at Illinois. And then kind of the stars of this talk is really Saprit, who was one of my first graduate students, Santosh, and Helwan, who is the most recent person to work on this, and also Lon Jubis, who's now kind of looking at similar questions in his own lab, and who is also a phenomenal student. And so hide my slides, and I'd be happy to answer any questions people have.
Thanks, Chris, so much for this outstanding talk. We really learned a lot about virulence regulation, especially in Salmonella, and really excited about your future experiments with the antimicrobials and its relation with virulence. So I have a couple of questions, but I will leave my questions to the end of the talk. Maybe any of the attendants can have any questions for Chris. You can type, and I also can narrate what's typed also.
So Dr. Mohammed, Dr. Rami raised his hand. You can see raised hands here.
Okay,
right.
Dr. Rami, so you can, the floor is yours, Dr. Rami.
Yeah. Thank you. Well, yeah, I have a very small question, but before that, I'm really amazed. It's one of the really clearest and best talks I have attended in molecular microbiology in general. Really impressive how it goes from the very basic basis of regulation to, I mean, I really enjoyed it like a musical piece because it flows really well, and especially the techniques used, because we have been now so used to not looking at cells, like just looking at gene expression on the RNA level, which is not exactly what happens in phenotype. So I really enjoyed it, and the reason I'm saying this also is I wish that the organizers will make this available. It's very educational for our graduate students, if they may. That's the comment, and my small question is, I mean, it's not, well, it's not a question rather, but an open one, and I'm sure you have answers to it, because I'm also a fan of antivirulence, like trying to attenuate virulence of bacteria, and so we can live in peace together. However, we all know that virulence is not intentional. If we want to say, like, bacteria don't really intend to harm the host, but they're mostly trying to evade our immune system, or they just have some waste products that happen to be toxins to us. So my question is, the balance, like the bacterial homeostasis and balance may require these systems that we are trying to knock down. And so if we keep the bacteria alive and just aim at removing their sensors or their flagella, for example, or whatever is directly involved in virulence, wouldn't this, I mean, it has a purpose for them, or like a selective advantage, wouldn't they try to evolve any other fitness factor that may be virulent? It's like you're still putting some dangerous person out of jail, for example, and or wouldn't they acquire resistance? I'm not like, it's a question that I ask to myself too, because the other side of the story is also not very safe, which is you kill the bacteria, so you're putting so much pressure on them and they evolve resistance. So, but what's your take on this? Like, you try to target systems that do not seem to be very essential in humans. Maybe they evolved in another animal, and so on.
Yeah, so first of all, that's a fantastic, well, first of all, thank you for the positive comments. I really appreciate that. That's actually a really complicated question that you asked. And, so I think, it's obviously, like if we take E. coli, for example, there's probiotic strains of E. coli, there's pathogenic strains of E. coli. They vary quite a bit And so, a non-pathogenic bacteria can be potentially beneficial to the host. It can also be negative to the host. I do agree that, if you're trying to downregulate, one thing, if we go after SPY1, it's possible that other mechanisms will evolve in those cells, and that will promote, them becoming pathogens again, and, negatively affecting both livestock and human health. I mean, salmonella is a big livestock infectious agent. I think, what's interesting, I think, about looking at this is it's just a different class of targets for, antibiotics. Typically most of it's focused on, cell wall biosynthesis, the ribosome, RNA polymerase in bacteria, which are different than eukaryotes. Here we're actually going after the factors that are affecting virulence, whether that's as good as, ampicillin or penicillin or tetracycline, I doubt it. But, it could be used to at least, reduce the strength of an infection in an otherwise antibiotic resistant strain. And so that's probably my best guess. I won't talk longer on it because this is one we could talk for hours on.
No, thank you so much.
Go back and forth. But great question. Be a nice one to have over a coffee for a couple hours.
Thanks, Chris, for the answer, and thank Dr. Rami for bringing this up. Dr. Ahmed Sharif has a question. So, Dr. Ahmed, the floor is yours. And then Dr. Amr Hanoura. Dr. Ahmed, تفضل.
Hi, thank you very much. Thank you for the organizers, and thank you for Chris for this very nice talk. I have a couple of questions here. So maybe I'm not very familiar with the salmonella, but have you tried to test your mutants and this effect in an animal model? I know that there is no one that will mimic the infection in human, but at least to take it to the in vivo part. And also I noticed you got a couple of mutants there in your target genes, Helwan. But I didn't see a complemented mutant, so I'm not sure how far the attenuation in expression is reflected to a mutant. So yeah, we can decrease the motility, you can decrease the flagella, but is it like a mutant, so it is totally unable to cause infection, or it is midway?
Yeah, so there's a couple questions there. Let me think if I remember them all. So the first question was about using kind of animals. Typically mice are the model for salmonella. It's actually a decent model for them. We haven't done any animal experiments. Part of the reason why is kind of the resolution that we require to measure gene expression. We couldn't get using an animal experiment. We have to grow this in vitro. And so that's a limitation on the experiment. That's also why we can't look at the systemic phase of infection. So there's a second process once they invade intestinal epithelial cells, then they go after macrophages. We just can't do the resolution of experiments in an animal host. So that's kind of one limitation. But a lot of people have tested these different mutants in animal hosts. So some of the regulators, like the master regulator, like Helwan, you knock that out. Basically, there's no infection by salmonella. Some of the other ones, you see a minor reduction in basically infection. Motility, if you knock it out, you see a significant reduction in kind of infection, just because the cells can't move, they can't penetrate the mucoid layer of the intestinal epithelia. So chemotaxis is actually not required for infection, it's just movement is required. And we know there's cross-regulation with gene expression. And then there was a technical question about complementation. Some of the genes we can complement. So all we do is targeted mutations. So we're actually going in and we're using what's known as lambda-red recombination to do targeted deletions. So we can sequence the mutants. So we know we have the deletions we want. We know there's no off-site mutations. So we have lots of controls for that. We have complemented some of the genes, but some of the other ones are really hard to complement because if you and particularly some of the Hill proteins that regulate SPY1. If you overexpress those proteins, like in a complementation experiment, you actually make too many needle complexes, which kill off the cell. So there's like you can't have expression too high because it'll kill the cell. And we've kind of struggled a little bit to do some of the complementation experiments. Everything in the flagella motility realm we can complement, and some of the ones in virulence we can complement, but others we can't, just because when we try complementing them, we kill off the cell. We're pretty confident about the mutants, but ideally we do the complementation experiment.
Thank you very much. Thank you.
Thanks, Dr. Ahmed. Thanks, Chris. Dr. Amr Hanoura.
Thank you very much for your presentation and your talk. It's a very good talk, and I like it very much. And I have a simple question. What is the interconnection of this network with the quorum sensing inhibitors and quorum sensing? Is there any connection between this network of motility and variance with quorum sensing and quorum sensing inhibitor?
I can only give you a partial answer because, to be honest, I forgot the details. Quorum sensing is definitely involved. And I forget whether it targets virulence or motility. It could do both. And I don't know, unfortunately, I don't remember the details off the top of my head. So we haven't looked at quorum sensing, but it's definitely involved. I will say there's also, you can argue this is quorum sensing to the degree. So I mentioned short chain fatty acids, right, are a signal that induces virulence. When you grow the cells, often they're going to do fermentation, and Salmonella will produce acetate. So just through its metabolism, it produces acetate. Acetate can affect gene regulation in these organisms, in particular virulence gene regulation. And so aside from like homoserine lactones and other kind of quorum sensing molecules, even just production of short chain fatty acids can also potentially be a quorum sensing molecule. But it's an area I haven't investigated, so I don't know. But it's definitely involved. And now I'm going to spend the afternoon digging up on quorum sensing.
Well, thank you so much. I think we have like more questions, so it seems like it was an interesting topic. That's why I like to have a lot of questions. So Shadi Zarif, so the floor is yours, and then Shahida Nofel. So Shadi's first.
Yes, thank you for this presentation. I have like a question, like you said that NAC is active with Syncytia at Dr. Amer lab, and when I work with NAC with Bronchitis and Syncytia, I like found the totally opposite phenomena. So when I add like NAC to this bacteria, I either take the host cell when I infect macrophage, NAC like as an antioxidant decrease like reactive oxygen species, which is attack so when I add NAC, I found increase in the growth of bacteria. So my question here is like Salmonella is affected by reactive oxygen species, and that's why NAC is act as antibacterial to Salmonella. Thank you so much.
Yeah, so basically what your observation is that when you have NAC present, it actually promotes the survival of bacteria within vacuoles or macrophages, because it's antioxidant and it prevents the production of reactive oxygen species that enables Salmonella to survive in Salmonella containing vacuoles within macrophages. That makes complete sense. There's been actually a lot of work on just the innate antioxidant mechanism in Salmonella. And that's actually a really good point you raised. So one of the things when we did the NAC experiments is this is just vegetative growth. So we don't have a eukaryotic host. We're not adding it at the moment of infection. So it's definitely inhibiting growth. But clearly, as you mentioned, it can also, under certain conditions, in the presence of, say, like hydrogen peroxide, like in a vacuole, it can actually promote growth. And I think that's really interesting, but for some reason, it's down-regulating the virulence genes. One possibility is that typically virulence is used, kind of the SPY1 genes and the motility genes that I talk about are used to kind of get into cells, but once they're inside the cell, they're going to turn off that machinery and express different machinery so they can survive within the vacuole. It's possible that NAC is, as an antioxidant, is down-regulating motility because it's making the cell, right? No, that's backwards. Let me think about for one sec. It seems to have, depending on the phase of growth is probably the best answer. So during vegetative growth, it's an inhibitory, and maybe during the infection process, it promotes survival, but it does down-regulate the expression of the virulence genes. And so that we've definitely seen. And a number of other antibiotics like salicylic acid, if we do like longer chain fatty acids like bile acid, paraquat, which is a redox cycler, those are all going to down-regulate it. So I think there's a lot more work to be done. That's probably the best answer I can give you. But thank you for sharing that. That was quite informative.
I think Shadi, I think Shahira is next. Shahira, I think you raised your hand. So do you have a question? Okay, so until Shahira is coming back, Chris, I have a couple of questions actually. The first question is what like triggered my interest here, because as I told you, I'm studying the evolution of antimicrobial resistance of different foodborne pathogens as well, using some genomic tools. So the question that triggered the hypothesis based about this correlation between virulence and antimicrobial resistance, that both traits are actually acquired during different time. Like the virulence, actually, as Dr. Rami mentioned, it's following colonization, for the bug to evade the immune response and to enhance colonization of the bug, maybe through acquisition of different genes, through the gene pool or so. Yet the antimicrobial resistance is through the selective pressure from using the antimicrobial resistance. So and I also found some actually antimicrobials are actually decreasing the virulence, yet others in different bugs and different antimicrobials are actually increasing the virulence, and other antimicrobials has no effect. So talking about the Salmonella, maybe as an example, do you think this actually existed in different pathogens? What is the evolution link between the antimicrobial resistance or the hypothesis? It's adapt or perish for sure, but what is the evolution mind map for both co-selection of virulent and the antimicrobial resistance strains? So this is my first question. My second question is, I think I also mentioned this about maybe you studied some kind of regulators for flagella, for motility, and for islands, etc. So Amr actually mentioned about are you interested also in some kind of global regulators such as the Curam sensing, or also anti-biofilm inhibition? Finding some kind of regulation of biofilm would be really a good target, especially in Salmonella or other biofilm forming organisms.
Yeah, so let me go backwards. I'll answer your second question and then your first question. So we actually started pretty small on this. So actually initially we actually had two separate independent projects. So one was looking at flagella regulation. The other one was looking at virulence regulation. And they were very myopic, just on the genes directly involved in that regulation. And then it's only later that we started looking at the interconnection between the two. And we started to understand them well enough that we could start thinking about kind of two systems. And then we've kind of gradually, slowly kind of increased the complexity of what we're looking at. So probably the most recent thing is Helwan's work, where we're looking at, response to short chain fatty acids, crosstalk between the two systems, and just the complexity of the responses. And there's still a lot of work to be done in that area. But yes, without a doubt, I think global regulators are, another target to look at. So there's just, it's just getting enough people and enough research funding to pay for it so I can do it. But there's a million questions to be answered. your first one, I think, is a really interesting question. And I can just tell you my perspective on it. So when I look at virulence, so in Salmonella, they typically estimate that probably 500 genes are involved in virulence. So probably 10% of the genome is involved in the virulence process. And from what we understand, the decision of a cell to express its virulence machinery or not express its virulence machinery is determined by all different kinds of factors, environmental factors, growth factors. And basically, we figured out ways to kind of tease the cells in the lab to express these that is separate from the mouse. So that allows us to do all the genetics, the detailed measures of gene expression, everything else. I think antibiotic resistance, at least my understanding, outside of general stress responses. So I'm not talking about general stress responses, but if you think about the acquisition of like a beta-lactamase, that's going to be acquired independently of the virulence machinery. But when you add, like a penicillin, or ampicillin or any other kind of antibiotic, it's going to affect the health of the cell. It's going to affect its general health, its general physiology. Even though it might be resistant to that mechanism, the ability to fight off the antibiotic is changing the metabolism and the state of the cell. And since the virulence is intimately connected to kind of the physiology of the cell, you can easily imagine scenarios. So we've talked about scenarios where that stress of an antibiotic is downregulating virulence, but you could also imagine cases, maybe not in Salmonella, but possibly that stress is actually going to increase virulence because maybe the cell is using a stress signal to know to express its virulence machinery. And so I think a lot of it's going to come down to just understanding the logic in these cells, to the ability that we can determine them. There's a reason why we can do these experiments in Salmonella, because it's easy to do genetics in Salmonella. If we were going to try to do this in, a Clostridial species or in Enterococcus faecalis, it would be infinitely more difficult. So there's some advantages to Salmonella. That's why we can get farther with it. So I think it's just complex. I don't think there's a general answer. I think it's going to be condition dependent. But I can tell Salmonella, because it's so integrated into the overall physiology of the cell, any stress is going to affect virulence one way or the other. I think it'll be difficult to predict. And I definitely think there's a lot of, I think quorum sensing is a really interesting area to pursue.
Oh, thanks, Chris, so much. I think that was so informative. And we are really interested in looking into it. Yes, Shahira is in, I know. Shahira, you fix your mic?
No. We have two questions, a couple of questions in the chat, Dr. Mohammed, one from Sahar and one from Shahira. She has a problem with the mic and she wrote the question.
Okay. So, okay, Dr. Lema. So, I think Sahar is mentioning some like a question. I'm gonna mention it. Why did some experiments using some supplements to downregulate quorum sensing genes in Pseudomonas, but really we found some of the real isolates was downregulated and others was upregulated, and they were of the same species. So, do you have any explanation for this?
No, but I think it's really interesting. And there's definitely a lot of differences between different isolates. I have a colleague who studies like urinary tract infections with E. coli, like pathogenic strains of E. coli or uropathogenic strains of E. coli, and he sees all kinds of variability. Most likely what it is it's probably mutations in the promoter or the master regulators or the global regulators. So typically where you see the variability is like, it could be the master regulator of motility, it could be the master regulator of virulence. And so that would be my best guess, but yeah, I wish I'd give you a better answer. I don't know the answer to it, but I mean, these are plastic networks in the sense that, you change the expression of one transcription factor, you affect the expression of 20 other transcription factors. So you can easily imagine cases due to the complexity of these circuits where a single mutation can flip the response that, in one case, as you mentioned, downregulating quorum sensing has an effect. In other cases, it doesn't have an effect or even makes things worse.
Thanks so much. Another question from Shahira. So she had a question about the small population that stayed non-motile even when they are even the nutrients were increased. Can you separate them from the rest of the population, then try and increase the nutrients again? So she's thinking maybe they represent a reservoir of cells just in case of expressing cells are depleted.
Yeah, we haven't done that experiment, but I'm almost, well, one, there's a technical issue with doing that experiment in that we're not allowed to use live cells in the flow cytometers to do sorting, and that's just due to biosafety concerns. And so right before we measure expression, we actually kill off our cells. We treat them with a lot of chloramphenicol because we can't have, so we actually have a pathogenic strain of salmonella, so we can't just have it on machines. So that's kind of a technical issue. That said, that population off, if we were actually to sort it out, I'm 95% convinced, can't be 100% because we haven't done the experiment, is it's just gonna look like regular bacteria. Because that population is too big to represent something that's genetically different. So I think if we grew out those cells, they would just come back again, and we again see 5% that are not motile. Helwan actually she did an experiment where she made basically like a hypermotile cell of salmonella. I guess technically it was hypervirulent too. And that basically we were able to get rid of that small population and everything's on. So it's definitely at the level of regulation. It's not, I think, a genetic mutation, but I'm not 100% convinced because we haven't done that cell experiment, and it would be really difficult for us to do that experiment just because the university is not going to allow me to run live salmonella through a flow cytometer.
Thanks, Chris. I can see any other questions. So again, I would like to thank you. I would second like what Dr. Rami mentioned, that personally, this is one of the best presentation that spans beyond the very basic 101 biology to the sophisticated molecular experiments. I really appreciate how you flow from the very simple concepts to the very sophisticated experiments. So I really appreciate that flow. Again, I also appreciate that this type of research is one of the innovative potential R&D experiments that have a potential to combat antimicrobial resistance. And as you know, most of the guests here are some kind of link to antimicrobial resistance, especially in Egypt, where the antimicrobial resistance rates are very high. For this, I would like to thank you again, and I will give the floor to Dr. Sameh to conclude our session.
Thank you, Dr. Mohammed. I would like just to thank Chris. Actually, it was really very interesting talk, and we have seen how lovely was the discussion and how people were engaged, and we have a large number of participants actually, and this is the essence of this Let's Talk webinar series actually. So thank you, Chris, and we hope we can host you once more in the future. I would like also to thank Dr. Mohamed El Hadidy for the excellent moderation, and I would like to thank all the participants for their engagement and their presence. And I would like to invite you all for our Let's Talk Science webinar in March, development kallikrein-related peptidase inhibitors. Our speaker is Professor Nicolas Mizior from Montpellier, and our moderator will be Dr. Ashraf Abadi from the German University in Cairo. I would like also to, at the end, to thank all the organizers and the team who helped me in the organization, Silwan, Chadi, Mahmoud, Ola, and everyone. Thank you all, and see you in March, inshallah.
Thank you, everyone, and thank you for the fantastic questions. Really insightful, and I really enjoyed it. And hopefully one day we can do this in person once the pandemic ends.
Well, actually, we'll be happy to host you here in Cairo.
I'd love to visit once I can leave the country without quarantining for a month. Thank you.
Thank you. Bye-bye.